Buonasera, benvenuti a
Good evening. Good evening and welcome to the 32nd edition of GiovedìScienza Science on a Thursday. I'd like to invite our guests this evening. My role is not like the usual one, but very amusing. First of all, you see that the stage now has many more people, and there are many more people because there's a new object here, and that is to say that gong over there. After 32 years, it was important to change conducer. We have Alberto Agliotti, whom we'll be introducing now, and also to introduce some changes, a few grains of sand in the clogs, because after 32 it was too easy. So I'm sorry that there are not very many people here. It's well, word went round that I was here, but we will try to tell you about what happened and then circulate it, because word of mouth is very important. We haven't got many tools of communication. They're all things that are very expensive that we can't afford. So we count a lot on word of mouth. So my task this evening is to explain what the gong is about and why there are two young people here that should have been sitting down and that were captured on the way and forced to sit next to Alberto Agliotti, two volunteers, so to say. These two forced volunteers have a task, and also a very difficult one. That is to say, they have to beat the gong. They have the right to one each, so it won't be too many of them, just one, with that object that you can see in Agliotta's hand. I don't know what it's called, but it will interrupt speakers, I hope with politeness, and you can interrupt them when if you feel there's something very important that you heard and you'd like to hear it again, or something that you didn't understand, and so you ask for an explanation, or C, there is something which raised a doubt, a problem, and in that case they will decide when, if, and how they will be playing the gong. Nearly finished. Alberto Agliotti, who was here on Sunday when the theater was full and there was Piero Angela, knows everything. And so this person will have to hear it again. He's an electrical engineer. He graduated from the Turin Polytechnic, then for 2 years, 1 year. He worked for his PhD. Let me say 2. In any case, I did not finish my PhD because Pierangelo didn't finish the Polytechnic, but then he did what he did. And in the end, he managed to work well in life. And he stopped because he wanted to play. Alberto Agliotti is also a musician. And the first instrument is the violin. He went to the music school, and then the second one was the saxophone, and then there were others too. I am both baritone and a tenor. I play them exactly in the same way, but don't crack a joke about that. What can I say? He established something which is called Occam's razors. Occam was a philosopher, as you know, from the Middle Ages, that had a very beautiful idea that he said that there are two explanations. One is complicated and one is simple. Probably the right one is the simple one, and you cut with a razor. He was a monk, amongst other things, but it was one way which is debatable, but it works in science. It has always worked well. Occam's razor does a lot of things, and one of the things he does is scientific communication. He's organized about 10 of them. And now I will talk about the bacteria of happiness, published by Herpley, which by a British biologist who's a scientific popularizer, Anna Cullen, hopefully the bacteria of happiness. This evening we'll hear people talk about bacteria, and these bacteria can contribute to our happiness in various ways. So a first conference which is well boding. I'll now give the floor to Alberto Agliotti, and I will take my chair away like he did, but I am not wearing an evening coat as he was on Sunday, and I will leave the floor to Alberto. First, he is going to interview very briefly our guests, and then we will move on to the serious part. Thank you.
Ringrazio Piero Bianucci per questa presentazione. Ringrazio Centroscienza.
I would like to thank Piero Bianucci for this presentation. I would like to thank the Centroscienza for this opportunity. And in particular, I would like to thank Piero Bianucci. I don't know if you two will have to thank him for this, but I would like to ask you to give him a round of applause. And now, let's move on to the most serious part. That is to say, we have with us...
Salve, sono Valentina Fossati, studio Scienze Naturali a Torino.
My name is Valentina Fossati. I study natural sciences in Turin, and I'm here because I was forced to. That's not what we had agreed to say, but I love science, biology, and so I hope that this conference is going to be very interesting. Good evening to you all. My name is Simona. As Valentina too, I am a natural science student. I'm about to graduate. I love animals. In fact, I work also with the dog pound in Turin, and I would and the dog shelters. I am here too because I was forced to. I would now like to try the gong, if you agree. Get up and hit it in the middle.
Credo che si senta, vero? Lo sentiranno.
I think you can hear it, can't you? Yes, they will certainly hear it. Our speakers, whom I'm about to give the floor to, Duccio Cavalieri and Carlotta de Filippo. They will be talking about bacteria, and not just bacteria. The first one to speak is Duccio Cavalieri, is a professor of microbiology at the University of Florence, and if you'll allow him, I would like to give a quotation of his, which he has on his website, which is by an Afro-American singer. So music is back, who used to say that the blues is to jazz just like yeast to bread. Without it would be flat. In the case of this evening, maybe we should turn it round, but I would like to give him the floor now.
Io non vi parlerò di batteri, vi parlerò di lieviti.
I won't be talking about bacteria, but I'll be talking about yeast, and I will be talking about a story which interests us all, both those of us who fermented cereals and those who don't. It's a very special story because they're looking for a link between yeast and the mycetes, which we have had for thousands of years in fermentation, and that have affected the relationship between our eating habits and nature. I don't know if you know what yeasts are, but yeasts are microorganisms which for the first time were observed at the beginning, middle of the 17th century with very, very primitive microscopes, but very effective to see yeast. Robert Hooke, 1660, was the first one, and you can see he was using the microscope on the right. With this, he was able to observe molds, as you can see on the right, and then Van Leeuwenhoek was a clothes and fabric merchant, and he saw bacteria and yeast, and he looked at beer. van Leeuwenhoek lived in Amsterdam, and so he had access to fermented beverages, and he looked at these microorganisms, which today you can see it with a more advanced microscope. You can see there's a scale of 5 micrometer, and the yeast goes between 2 and 5 micrometers, and the ones you can see inside are both cells per se and also ascomycetes. It is the product of meiosis, which are gametes. This is fundamental information because this is the microorganism where for the first we were able to study the development of sex. So yeast have a form of sexuality, 2 types, and have a cycle, sexth cycle, which is very complete and very interesting because it generates diversity. But we discovered it in a lab, and until a few years ago, no one had the faintest idea of where it happened in nature and why this process had developed. This is the image of the moon, that the craters of the moon, which we will be talking about later. This is a Petri dish with yeast scattered on it, and these are the shapes that they take. The reason why many years ago, 24 years ago, I started to study the cerevisiae, because I was working the Mario Puccinelli lab, he's one of the founders of Italian microgenetics, is that we use 2 plates. So the Escherichia coli, reminded me when I went to the bathroom, while the Saccharomycetes reminded me with the smells my grandmother's house. So when I had to choose one, I chose the yeast because it reminded me something ancient, which was very strongly in my ancestral memory, and that is to say, that's a smell. Now, the relationship between yeast and science is a very ancient story. In 1680, as I showed you, Anton van Leeuwenhoek observed beer yeast the first time. In 1837, Cagnard-Latour showed that yeast is responsible for fermentation. In 1839, Justus van Liebig and Frigger-Wöhler showed that alcohol is produced by yeast, and this is the wonderful humanization of the yeast cell. Cagnard-Latour imagined that yeasts were like small little animals that sipped sugar through its snout and excretes alcohol from its gut. Imagine that there was a sort of a gut and also emitted carbonic acid from its urinary organ. So alcohol would be the feces. And since we'll be talking, or the next speaker will be talking about the digestive system, I thought it would be quite nice showing the comparison. In this conversation, I would like to, in fact, refer to giants in the science. Without the work done by Louis Pasteur and by Emil Christian Hansen, probably this part of human science would not be where it is nowadays, because the former, the first one to understand the central role of yeast is Louis Pasteur. In 1876, he published Études sur la levure de la bière. And in 1874, Jacobson, which is the founder of Carlsberg, Carlsberg means the hill of Karl, and that's where he lived. He was a professor of the University of Copenhagen, and he asked him to go and work with him because he had to stabilize a yeast which was fundamental, which then became Carlsberg's beer. Christian Hansen isolated them in pure culture, stabilized them, maintained them, and then he started to study its biochemistry, and had studied the first enzymes from cells of Saccharomycetes. He studied and resolved one of the enigmas but I'm sure of our politicians worry about, because in 1871, Pasteur gave the first and I think the best of the difference between fundamental and applied research. He said that there was no category in science which can be defined as applied science. There is science and the applications of the science, and these are linked the one to the other, just as the fruits of a tree that bears it. So a healthy, good tree which we have invested on will give good fruit. A bad tree will give very bad fruit. And I will leave it to you to judge the tree of our science, how it was being nourished. Yeast has been unquestionably the best model to study in genetics and genomics, and it opened a new era of biology. Very few people know that the first links to DNA or studies on DNA were in fact studied in the Saccharomyces and Cerevisiae model. If we have a scientist who is called the Beadle Medal for Genetics, was awarded as like a Nobel Prize. He'd worked in World War II on with the American troops in Japan, and used yeast to study the effects of radiation, the radiations which had been triggered by the atomic bomb in Hiroshima and Nagasaki. And looking at this, he cloned the first genes that are responsible for this, very important genes, for example, for the study of cancer. Bob, in his life, he mapped the 6,284 genes of the Saccharomyces cerevisiae on the yeast. In 1986, when yeast was the first genome to be sequenced, there was a 95% match with what this great man had done, and the position of the yeast. I was the last PhD student of Bob Mortimer's, and that was a privilege. As we all know, here are the interaction between bread and wine, and this was the basis of the Nobel Prize in Physiology for Medicine 2001, because yeast were studied for the cell cycle and the progress of the mechanisms that develop hereafter. And recently, yeast gave another Nobel Prize for Biology. This is 2017, Yoshinori Ono. I was present in Japan when he gave his Nobel laureate lecture, and he said this sentence which really surprised me very much, which has to do with the fact that the seeds of great discoveries are constantly with us, but in fact they yield fruit only in the minds that are prepared to receive them. This is the task that many of us have. That is to say, we have to prepare our minds and the minds of our students. My story with the Cerevisiae starts many years ago. I worked 6 years in Harvard, and my NIH's research project was called In Vino Veritas. That was the logo. You can see it on the right. That refers to a very ancient Roman motto. And we worked, and this was one of the first jobs, we extracted DNA from a very ancient finding from the Temple Valley, from Abydos in Egypt. These were vessels where Pat McGovern, who is virtually the father of the study of ancient fermentation, had found a red material that looked at wine. Chemical analysis said it was wine. We sequenced the DNA, and we discovered the Saccharomyces scorpionis. We gave the name to the ancestor of the Saccharomyces cerevisiae. This was really a very ancient history. I think very strongly that the Saccharomyces cerevisiae and its product are part of our cultural being, because the story of beer dates back to the Sumers in Egypt, India, China, and Homer give us a very important reason why the fermented products are important for our culture. He says no poem was ever written by a water drinker. He says this, the person who writes the Odyssey and the Iliad, and a very ancient Egyptian proverb used to say in vino veritas, because you see in water you see your own face, but in white you see the heart of its garden. The other thing that very few of us think is that what beer is, what is wine. I am going to give you a neo-Darwinian interpretation. I don't believe in the natural selection per se, but this was published by a few colleagues. It was called Ecology, Tribal Warfare Maintains Microbial Diversity. Wine and beer are the fight between the yeast and the microbes, because yeast divides itself or reproduces takes it 4 times as long to separate and to reproduce as it does bacteria. So if it wants to survive, it has to use a strategy that many human beings have learned how to use. That is to say that you have to be an attacker. Instead of running faster or using resources at the best, it develops weapons to eliminate the others and therefore take resources by fermenting sugars. Saccharomyces cerevisiae produces ethanol and acetic acid. The production of these toxins is expensive because in practice it has 1/20 of the energy that it could obtain if it breathed, in fact, or fermented. So it sacrifices 19 parts of energy to produce toxins. Clearly, the ability to produce this toxin is auto-limited by the capacity to resist. The incredible thing is...
Mi scusi, la mia domanda non è proprio inerente a questa.
Please excuse me, my question does not refer exactly to this, but before talking about yeasts, etc., could you go back to evolution? That is to say, where they come from a prokaryote to a eukaryote cell. Yeasts have an origin. Well, it was defined as a unicellular organism. We thought that this was a very ancient trait, while yeast probably has extremely evolved. The genome is very compact. It's a perfect, near perfect genome, and probably the fact that it's a single cell one is a point of arrival. It went from being a multicellular like mycetes and molds. Clearly, after the bacteria, we have mycetes, but we have complex being. But fungi, by no means, can be defined primitive organisms. In fact, the genome, there are no introns here, and this means that it simplifies to the umpteenth degree its situation. May I continue? Thank you.
Questa capacità di produrre tossine.
For years, we used this ability to produce toxin to do something very simple. That is to say, to make some products of the farming industry preservable. For example, grapes, because when fermented it can be stored as a wine, but also as malt, for example. Because if fermented, we can keep it as beer. For example, it's like liquid bread. So fermentation is one way. That was true also for barley, for example. The Romans used the wine and beer to make water drinkable. Caesar and the Roman legions brought wine with them, because diluting wine 1 to 3 with water, they managed to make all water drinkable. Some people, for example, do it. They used it with water that had salmonella and diluted the 1 to 4, and after 40 minutes, salmonella was 0. So basically, it was one way to guaranteed drinking water. I left a trace on the yeast. This was published by Gianni Lita and his group. He's a very good Italian scientist working in France, and basically they saw that the strains of the wine are very similar, so I don't know if it's domestication, but certainly there is an interplay between the yeast industry and human beings. Where does this become very interesting? When we talk about domestication, it means that you're talking about the relationship between a world of natural yeast and human-related yeast. By the end of the 90s, we understood that Cerevisiae was not a domesticated species, but that there were strains that had had a relationship with human beings and probably revolved through clonal amplification, and others that were natural ones. The natural ones were linked to trees, while the industrial ones to wine. And there was this hypothesis, which was the Gondwana hypothesis, using genetics to follow the flows of yeast, as if we were following populations. And you can see there's a flow of yeast from China, probably, in fact, from Mongolia, Afghanistan, right to the Mediterranean, and then from the Mediterranean to the New World. This was the Gondwana hypothesis because it simulates or it follows the movement of continents. One of the big problems is how yeast went from one place to the other. I wondered for a long time, how did this happen? Because they don't have any legs, and many, in fact, in the part of other organisms or microorganisms are carried by wind. But Saccharomyces cerevisiae is heavy, very heavy, and it can't. So it can't be moved by wind. So the question which I had a few years ago is how do yeasts move? Where do they go? And how do you explain one of the big paradoxes that virtually blocked microbiology of yeast for years? That is to say, on grapes, if we go to September, only 0.5% of the grapes have Saccharomyces. But if I take it later, then in the autumn, then you have virtually 25%. Where does it come from? The trees, from the environment? What is the vector? This was the answer. This image, which I took a few years ago, and I started developing the hypothesis on who the vector was. This is a hornet, which is eating a grape which has withered, and you can see that there's also an ant. And so I started thinking that the vectors had to be insects, and testing the ability of them to break the skin of the grape, and we discovered that only the wasp was able to move the Saccharomyces. The other thing of the Crabro wasp was practically matching to when you started to isolate some strains of Saccharomyces. That is, if I went into the wood or I went into the vineyard, from July I could do it, while from November it was extremely rare. And this is exactly the cycle of this wasp. From their nest, and then there's the sexual phase, and then the hibernation. So we carried out a metagenomic analysis, and I'm sure that the Professor De Filippi will explain this better. We took the animals, we dissected them, we looked at the two parts of the intestine, we identified the organism, and then we took the intestine, the gut, and we sequenced the entire DNA. We saw different types of fungi, and we found more Cerevisiae before the ripening of the grapes than after. But observation is not enough in science. It's necessary but not sufficient. So one of the things that we started to do is that we transformed our lab on insects. We nourished. This is a wasp. You see, we fed it. It's not a hornet, it's a wasp, because the hornets are rather dangerous, so we did the experimental side, was carried out on wasps. We nourished them with a liquid that contained the yeast, and then we checked if it still existed after hibernation in their gut. So practically, we used fluorescent yeast, and we saw that it was a GFP-labeled Cerevisiae, and after 3 months of hibernation, you could still find it. If you waited more, then the cells started to die. So after 3 or 4 months, there was a form of death of these cells. The other thing we wondered whether the queen could pass the Saccharomyces to the larvae and to the workers. So we took 4 nests. We nourished them with the same system, with the fluorescent Cerevisiae cells, and the result is that in 90% of the cases, there was a transmission of the fungus from the queen wasp to the larvae, and this too was interesting. It was a dispersal. Other researchers, or at the same time, while we were working on the Drosophila, they were working on the Drosophila, found the deletion of a gene in yeast, which is the anti-F1, which is promotes acetyl acetate. If that gene was there, the Drosophila was very interested in yeast. They eat yeast. That is why it's not a vector, because it digests it. But if I destroy that gene, at that point, the Drosophila is no longer interested. So one of the things that we started to do was to try and see if of the 466 volatile aromas and of the 644 for wine and the 178 of difference that are produced by Saccharomyces had some role in attracting insects. We haven't got the conclusive data, but the answer is basically yes. The insects like the aromas that we like too, because they make them attractive as a way to tell the brain of the insect, this is good. I am good. Take me with you. So it is a way to hitchhike, to be carried or ferried from the trees to the grapes. This is a hornet nest that was built within a tree, and don't forget that the Gondwana idea is that the wild Cerevisiae one are on the trees, while the domesticated ones are in our cellars. The metagenomic revolution is something I won't be talking about, but it will lead it to DNA, and by sequencing the entire genome of certain strains of wasp, we find that biodiversity of wasps reflects all the biodiversity. That is to say that they carry this. The yeast of beer, of wine, of trees from Japan, Africa. But the interesting thing, again, that 10% of the isolated strains were, in fact, hybrids between Cerevisiae and Paradoxus and others too. This means that we have to take strains of Cerevisiae with markers and cross them with Bianus and Paradoxus, the other two types. And the difference between Cerevisiae and Bianus is the same difference between humans and mice, and with Paradoxus between a human being and a rat. So quite interesting. In the guts of the wasp, we find hybrids of the same species or different species, but when you can see the pink purpley one is the hybrids, but when you keep in the gut the yeast for a long time, and that's the blue one, you do no longer find hybrids of the same species, but only hybrids between individuals of different species. This is very interesting because the other thing that we discovered is that Saccharomyces paradoxus, one of the tested species, could not grow alone within the guts of the hornets, but it managed to survive only as a hybrid. This is a very beautiful example because in practice it is an example that human beings discovered when they had to develop mules. In fact, the wasp is the locus, the place where you can generate biodiversity, and makes it possible for the yeast to survive in negative situations when winters are very long and the yeast has to survive in hornets, it can only do it if it uses the fitness of the hybrid. This is not by chance, because beer yeast or many lager yeasts are either hybrids or have a mosaic genome. That is to say, which has ancient traces of hybridization, and in fact not by chance. Beer originally is from the Munich area and in Denmark, that is to say, very, very cold areas. And lastly, I'll be talking for the next meeting. This is not unique, going from insects to human beings. We're finding yeast also in the guts of human beings, but the interesting thing is that within the guts of the human beings that has absorbed them for years, the yeast does not hybridize. And this requires the hibernation of the insect during the winter. This process, which happens in nature and has been happening for millions of years, and which associates very closely insects to the microorganism that we are bound to for thousands of years for fermentation. Now, with this I have finished. I would just like to recall that in this world where we talk about microbiota and insects, there are passengers and there are colonizers and there are vehicles. Hardly any passengers have harmed us. Many of the microorganisms have become colonizers and have become invasive. So the fact that yeast and other microorganisms have a limited lifespan in ourselves is one of the reasons probably why it is good, which is something I'd like to state. And lastly, I would like to close with this sentence, which is I think of every morning when I start working. Nothing in biology is meaningful unless read in the light of the evolution, and that's Dobozansky, and that the host of the microism that live in it are the unit of Selevg and Zibi and Leona Rosenberg. That is the basis of the microbiota, which is going to be explained now by the next speaker. Thank you.
La parola a Carlotta De Filippo.
I would now like to leave the floor to Carlotta De Filippo, who is a researcher at the National Research Center, CNR, in Pisa, at the Institute of Agrarian Biology and Microbiology.
Introdotto appunto il concetto di
Good evening to you all. Duccio has introduced the notion of microorganism and of the relationship which exists with animal organism. And I will be talking about, I will be taking you to Africa, where we have found some very interesting things in seeing where we come from, not so much our genome, but the guests that are with us every day and with which we share this time on Earth. In the last century, research focused on the pathogenic role of microbes. So this is a book by De Kruif, Microbe Hunters, and talks about the first years of medicine when it was looking for microbes and fighting them. If you read this book, you can see that there is the relationship between microorganism and human beings as a war, an ongoing war, rather than as a vision of mutual benefit. While today the things have changed, the view has changed. This is the message which I wanted to launch this evening. That is to say that today we want to go and see what the beneficial role can be and how we can maintain a balance with between microorganisms and hosts, and with the hosts to maintain health. Who are we? Well, we have to think that for every human cell, we have about 10 bacteria. In fact, 2 kilos of our weight, say if there's a human being who weighs about 70 kilos, bears about 2 kilograms of bacteria. So you must always think that when you go and weigh yourself, in any case, about 1.5 kilos or 2 are bacteria. But bacteria and microorganisms and yeasts, yeasts in our gut are about 1.2%. Most of the microorganisms that are in our gut are bacteria. Bacteria also have functions. In fact, if we go and compare our genome, that is to say, how many genes human beings have, with the genes that are borne by the microorganisms that we bear with us every day, we see that we're a minority, 30,000 genes as opposed to 1 million. This is why we talk about the human metagenome, that is to say, the sum of human genes and the billions of microorganisms. Currently, thanks to the technology, the advanced technologies of sequencing, this new world, which previously was unknown, has in fact become the cover of The Economist. Here you can see the Vitruvian Man that is described as a series of various species of bacteria. And there have been a lot of efforts on an international level to map all the microorganisms that populate the various parts of our body. As you can see here in this panel, we have all the microorganisms which live in our district skin, and then the oral cavity, the gastrointestinal tract, and the urogenital roots. You see that how each part, each role has a very specific ecosystem that favors health. When you have an unbalance, it's because there's a prevalence of one or the other, and at that point, there might be an onset of various diseases, and we'll be talking about it. I was saying that the advent of these new technologies of the sequencing, next generation sequencing, that makes it possible for us to go beyond the limit which due to the fact that most of these organisms cannot be grown. If we consider all the bacteria on the Earth, we can isolate and grow just under 2%, 1%. While what we're doing is that we're going to see the trace. It is what you do also with forensic medicine. That is to say, you go and seek traces of the presence. In the case of forensic medicine, you know, you look for human traces on the crime scene, but what we do is precisely this. That is to say, we seek the presence of bacteria through the DNA. Now, going back to that notion of symbiosis between bacteria and animal organism, because I'm talking about human beings because we're most interested in human beings, but in fact, this type of symbiosis is shared with all animal species, and I will show you this. What are the advantages of the gut microbiota? Well, the development of gut mucosa, so the children are born sterile. The contact with the mother's microflora leads to a first colonization, and this favors the development of the intestinal mucosa. It's a source of energy and nutritional substances such as vitamin K, which is the one which is important for blood coagulation, would not be synthesized. Then we have the products of fermentation of fibers, and we'll be talking about it because this journey to Africa helped us understand how the symbiosis between bacteria able to digest fibers that release a number of substances that are essential for our health. And then it's a barrier against pathogenous microorganisms because it educates our immune system. And we'll be talking about this later, because being too hygienic, too clean means that our immune system is not educated enough and therefore is not able to tell what is good from bad, and hence the triggering of the so-called self-immune pathologies.
Lei ha detto che solo una piccolissima percentuale.
You said that only a very small percentage of the bacteria can be grown in labs. Why is it only a small part and the others? Because most bacteria are anaerobic, all, for example, the ones in the gut, only 15–20% are able to tolerate oxygen, so much so that when we have the so-called coprocultures, what we go and see is a very low percentage of bacteria that are the ones that we can grow in the presence of oxygen. Most bacteria are anaerobic, and this means that in contact with oxygen they die. And the problem in this case is the sampling, because we cannot avoid very often the contact with the oxygen, and therefore we're no longer able to, in practice we kill them with oxygen. So thanks to this type of technology, we can map 100% of the bacteria. With the coproculture, for example, with the feces, if I have limited information on 15–20% of the bacteria, but with the DNA analysis,
È segno di successo. Senza nulla
No, perché avevo letto su un
it's a
articolo
sign of
che avevano
success.
trovato una
I'd read
differenza.
in an article that they had found a difference in the bacterial flora if you compared children born with a cesarean and children born naturally,
Sono più soggetti a problemi.
and that the children who've had born with a cesarean section are more at risk. This is a problem which emerged very recently, because what happens is that the first contact during the fact that the child goes through the vaginal canal, because all the lactobacilli, for example, are in the vaginal canal, and also with the feces during birth. And what happens is that during the cesarean section, you bypass completely this first inoculation, so you have a delay in colonization. In fact, there is a study, and the children who were cesarean sections have a lack or a very limited number of bacteria in the first weeks of their lives. In fact, and this will possibly be surprised, they're thinking in the US of putting the children who will have to have a C-section to have sort of to use the feces of the mother and vaginal bacteria and put them in contact with the child's body, so that the children will have that initial inoculation, which is absolutely necessary.
Ecco qui, come vedete, come capite,
Now,
inizia
here you can see this is the long journey which will take us to Africa, and the questions which are emerging, because what is happening is that in the Western world, autoimmune diseases are becoming more and more important, and they're also becoming quite prevalent amongst children. And so there's an idea that industrialization that led to processed food, use of antibiotics and other drugs, and also to more hygiene, have influenced our ancestral relationship with microorganisms. This gentleman, Graham Rook, was the first one who suggested that hygiene might be responsible for it, using also David Stratham's observation in 1989 that observed that hay fever is rare in second or third borns because they probably receive some sort of protection from their, quote, dirty, grubby older brothers. We don't know about this because the use of antibiotics is necessary when we are faced with an infection, just as hygiene is important. But what we're losing is that contact, that natural contact with the settings our grandparents were usually in contact with. The other thing is that the loss of fermented nutritional elements. There are a series of regulations that has limited the use of fermented food, and this is an impoverishment, while we see that in other countries this type of fermented food, which contain their natural flora, is a resource that we have to save. Here you can see, it's quite surprising, you can see on the left you have the incidence of autoimmune disorders, which involves the Western world, while we have a situation which is the opposite as far as the helminth infections, that is to say, worms. So we have seen or hypothesized that there is a possible influence on the environment of the onset of autoimmune diseases, because certainly there will be a number of genetic factors, but the fact that autoimmune diseases are not found in the developing world is clearly something that has to help us understand and try and find solutions. In this graph, you can see that if on the one hand we have reduced the incidence of infectious diseases, TB for instance, there is no longer a problem as it was in the 60s, but now you see that the increase of the autoimmune diseases is becoming an emergency. So we have to consider ourselves as a being that has to be in contact with microorganisms, which come from food and from the environment. Clearly, the more processed the food, the less we will have this cohabitation. The evolution of human diet, as you can see from this slide, we went from being hunter-gatherers in the Paleolithic to being farmers in the Neolithic. In this graph, means that we've gone from gathering and hunting, and then we've had more fishing and more hunting. And then during the revolution of the Neolithic, there were more farmers and more animal husbandry, in particular for grains, and this has meant that there has been an increase of them in our diet. What I would like to stress is that our day and age has a great use of prefabricated meals. The Neolithic Revolution was the first revolution that led to civilization. So there has been a transition from an economy of survival, which was based on hunter-gathering, to the domestication of plants and animals, with major consequences on human nutrition and on the social structure of the community, from the small nomadic communities to resident sedentary communities, and thereafter there was the increase of the density of the population, and then diseases started to spread, especially infectious diseases. Currently, we are monitoring them, at least in Western countries, but as I was saying, We also have a number of diseases such as allergies, obesity, diabetes, autoimmune diseases, chronic inflammatory diseases, and of the gut, which are also becoming a pediatric problem, that is to say, affecting children. The question is, how can diet influence this very delicate balance of the gut microbiota? To understand this, we went to Burkina Faso, to one of the poorest countries in Africa, and in particular to a rural village where, as you can see, it seems as if time had stopped 10,000 years ago. This woman, and you see it in the next slide, is still grounding grains as was done in the Neolithic. As you can see in the slide, there are these grinding stones which were used, so they're in museums, but here in Burkina Faso they're still being used to grind grains and produce millet and sorghum flour. Here you can see they have millet farming. This is one of the children who took part in the study, and in particular they have sheep, but they don't eat a lot of meat. In particular, they use them to sell it, and these are the so-called barrages, that is to say, water ponds where they drink from. So it's rather unusual, and I'll say why. These are the women that are the ones that farm with the help of children, and here you can see these stone structures where they grind millet and sorghum. This is an image which is quite suggestive of how this is the main meal, because this flour is used to make a sort of porridge which is called toma, and that's the male meal. We organized some questionnaires. It's not like the ones that we are more used to when a dietitian tells us what we should eat. But here you can see we managed to quantify the nutritional input of the various foods. It's mainly vegetarian diet, lots of fibers. These are the main ingredients which we analyzed. We analyzed the children because that's what we were interested in, to try and understand what the evolution of the microbiota is in the first few years. And the protein sources are basically pulses, which they call yabe. And this is a diet which is very, very simple. Now, thanks to techniques, sequencing techniques, which I mentioned before, we then went to consider the difference between the microbiota, the gut microbiota, between 14 children who lived there and 15 children who lived in a city which could be like Turin, but in this case they were from Florence. We were expecting a major difference because the environments are very different, and someone can say, of course, they live in very areas that are very different. They're exposed to an environment, to a flora which is very different, and therefore their microbiota is very different. Yes, on the one hand, but it's not exactly like that, because as you will see, diet has a key role. In fact, we found a prevalence of bacteria that digest fibers. Here you can see it, the Solanibacter, and the name says it is from wood. That is to say, they have bacteria that are able to digest also wood fibers. To tell you how powerful and the symbiotic power of these organisms. And our hypothesis was that is it possible that in this sort of environment, with this type of diet that is so poor of energy, because you have to think that to have 1,000 calories for these children is really complicated every day. They don't maximum, they get about 700 kilocalories a day. Is it possible that they manage to give produce metabolites that the human organism then can use? And the answer was yes, because we dose the short-chain fatty acids that come from bacterial fermentation, and you see that in the children of Burkina Faso the levels are very high. These are very powerful anti-inflammatories, and this is what happens when we eat fibers. That is to say, when we eat fibers, we select bacteria that are able to select them, and that then turn them into fatty acid, such as the various acids that are illustrated here. And this was a very important fact. Here you can see we try to organize it by clusters to see how far the Italian children were from the African ones. We saw that the children that were over 2 and that were not nursed, breastfed, were completely different. While there was a cluster, that was a central cluster with the 3 children from Burkina Faso and 2 children from Florence, Italian children, who had a microbiota that was very similar. So in spite of the fact that these children lived in environments that were completely different, their microbiota was very similar. We saw, in fact, that they had a lot of bifidus, and what was their thing in common? That they had been breastfed, or were being breastfed. It means that as long as we're being breastfed, we have a microbiota which our mother gives us because they pass through mother's milk. But when you start eating, then we select the bacteria according to the type of nutrition that we received. The bifidus remains in the Italian population because we use milk and dairy products, while in the case of the children of Burkina Faso, dairy products are not part of the diet, and so it disappears completely. And then we continued this type of research to try and understand if the effect of urbanization, the impact of urbanization, when these populations go from the rural village and move to a small city, here you can see Nanaro, and then move, in fact, to the capital, which is Ouagadougou, which is the capital of an African country, where they have food stuff which is more refined, as compared to the rural populations. We see that the situation changes. So you will observe that when they go from a rural environment to the urban one, we have an increase of calories per day. We have a reduction of the fibers, because clearly you introduce animal proteins or sugars, simple sugars, because on the one hand you have more energy, but you have a reduction in the number of fibers. So what we've seen is that, and here the dots are the various samples, and these are the children that we sampled, the feces of the children, and you will observe that you go from green to red and yellow and blue. So the process of urbanization in practice shifts everything towards the Western-like microbiota. The children who live in the capital, and we have interviews, eat cheese spreads with bread, and their microbiota is very similar to our children's. And clearly, this goes together with fatty acids, which decrease in this process of urbanization and change of diet. So what did we learn from this type of study? That diet has a fundamental, a key role in determining the type of bacteria that we have in our gut. And what can we do? Well, to safeguard our microbiota, what we must do is to maintain biodiversity as far as possible, because the more the microbiota is like a natural environment. You have to maintain biodiversity, woodland, rivers, and so on, but you must also maintain biodiversity amongst our microbes. And clearly, a increasingly standardized diet is not the solution. So you have to think of a microbiota as an ecosystem that has to be preserved. An antibiotic, for example, is a devastation. If you leave it, then you will have weeds, and the possible solutions can be the introduction of probiotics, which are the so-called enzymes and the prebiotics, that is to say, fibers which select the positive bacteria, and then the last one, which is a bacterial treatment or a fecal transplantation. Only in certain type of pathology you transplant a feces in patients, and therefore you use it as a sort of transplant. You use, instead of using chemotherapy, you use an antibiotic, and then you introduce a healthy microbiota which will repopulate it. And with this, I would like to thank you, and the journey has just begun. Thank you very much.
Allora, ringraziamo anche Carlotta De Filippo, ringraziamo le due nostre volontarie.
I'd like to thank Carlotta De Filippo. I would like to thank our two volunteers.
Alla
Thank you
prossima,
very much,
potete
Simona and
ricandidarvi
Valentina.
per questo
See you
ruolo.
next time.
Il mio era un gentile invito.
And once again, if you would like to
nascosto
have this
lasciare il
role,
palco,
I would
non volevo
invite
essere
you to
esplicito.
leave the
Eravamo d'accordo
stage.
così dall'inizio. Spero di non essere stato maleducato, ma invito a
We
tornare
had
sul
agreed
palco,
about
Piero
this
Bianucci,
since the beginning, but I would like to recall Piero Bianucci and also Teo Lusso.
produttore di birra, si può dire.
Teo Musso
Diciamo il nome della birra per
is
non fare
a
pubblicità, ma diciamo
beer
che la sede storica è
producer. We
a Piozzo.
won't say the name of the beer,
Come parlare di una fabbrica di
but it's in
motori
Piozzo.
turismo, pretendere che
It's as if I were talking about a Turin car manufacturer, not give the name. But you stayed in Italy, unlike the others, so I'd like him to talk about something about beer and yeasts. So,
Beh,
a lot of
grazie.
surprises today.
Allora, io sono qua in veste
Thank
non
you
di
very
uno
much.
scienziato nel modo neanche di un ricercatore, ma semplicemente di
I'm not a scientist or a researcher, but I simply work with yeast day in, day out.
un rapporto.
I live with them. I cohabitate with the yeast. A very good relationship. Sometimes it's a bit conflictual because they don't do exactly what I would like them to do, but it's a very beautiful side to it. In my life, I personally have followed about 3,700 fermentations. So if I have to say the truth, if we relate it to what is normally what you do, say, wine producers, we have 3 a year, with about 1,000 years I'd have to live. But with beer, you do it more often. All this world of fermentation, which is part of my life, I have thought many things. One of the things that I have thought, and which I worked for a while, because I hadn't understood anything at that time, was that I had to work for a few years where I symbolically put, but it wasn't symbolical because I used to put a lot of music because I wanted to see if the yeast reacted to music, listening to different sound vibration. I didn't understand anything, so I stopped that. After a while, I was lucky enough to meet Duccio because of something which was done with Pat McGovern, this great researcher, American researcher, and we worked on ancient beer, which was Etruscan-like beer. And the fact that we were together and talking, I was able to involve them, and we transformed this idea which music could influence yeast and the metabolism of yeast. We did some very interesting work which we haven't published yet, but which is very, very interesting. And which confirm my theory, not so romantic as I thought, but in my head I'm very close to that drawing where I see the cell of yeast cell as if it were a human being. I would have also added ears to it. I think there were a couple of bits missing in that picture that you had of that cell as a humanoid. And you didn't speak about the esters, which is the sweat. It's how that cell sweats, very important, because in the end, for those who have to produce something like I do, if you don't have that aromatic, if you don't have them sweating, then I haven't got any product. That's the part I'm interested in. That is to say that I have to give an aromatic profile to my beers. All this to say that, in my opinion, the fundamental key research will help us generate new worlds in our everyday life. And for those like me who manufacture, I'm not going to stop with Duccio. We have also started something very interesting, much closer to what he presented today, which is that discovery which is linked to the world of wasps and hornets. What do they do in the end? A sort of genetic mutation during the winter hibernation. So we used these wasps and hornets to map Italy, looking for wild yeast, and now we're trying them out in our labs in the Biotso beer manufacturing center, the brewery, where we have quite a good lab too. And we test these yeast strains that we found in Italy, trying to see what the reaction is, and we selected, there are over 60 that the University of Florence with Duccio has found in Italy, and we're testing them with the idea of finding some that are interesting enough, and we've already found something which is interesting, to putting them back in the gut of the wasps and hornets to rehybridize them and create a new strain of yeast that may represent Italy, a new Italian beer. It's a lovely objective, and we're working on it, and we're hoping to be successful. Thank you. Does Duccio want to add anything? Does Duccio Cavalieri? I wanted to say that I'll change the slide and I'll put headsets, because this idea of sweat is very interesting. Yes, I think that this is rather incredible parallel when I was talking about evolution, talk about the evolution of our senses. Insects have taste. There are things they like and there are things that they don't like. And there are flavors that are not only specific, but for example, the queens like different things from workers. I would not say anything here. I don't want to introduce a class slant to it, but it's quite interesting that what power determines, because I haven't said it, but the yeast is something which, not in my lab, but on a world level, we have deleted each of the 6,248 genes, which means that in the world of aromas, we can selectively delete this biodiversity, which we talk about can be used both to better understand the biology of the system, but also to give new opportunities or create new opportunities. Because when I put that sentence up where I wrote at a certain point that yeasts are part of our cultural heritage, I think that is something we don't think about. We think of a cultural heritage as our sculpture, our monuments, but The Gondwana hypothesis says that ever since the Romans started planting vineyards, when we started to use the fermented wheat to produce beer, we have enriched certain areas of a cultural heritage, a human cultural heritage. If you go to Chianti, the Chianti area, because you know it's ersatz. It is an ersatz environment. That is to say, it gives us opportunities that other people in the world have not got in that area. So the fact that you will preserve that cultural heritage is as important as the Uffizi in Florence or the Turin Archaeological Museum. And this is something which I think with Theo we share. I think it's a very important perspective. The fact that it is an invisible heritage, it doesn't mean that it doesn't exist. Electromagnetic waves from our smartphones are also invisible but still exist. I still wanted to add something. I think that leaving aside the work and emotional bonds that we established with Duccio, I also wanted to stress another point. The work that I tried to develop on 31 years, because as I've been soaking in beer for 31 years, is also the idea of a new vision of beer, which is closer to what Carlotta was talking about, in that I launched a notion of live beer. Live beer means that instead of beering corpses in a bottle, which is what industrial beer is, because it's cooked and completely killed. I mean, the live element is completely... the fact that you relaunch a beer where the yeast is still alive and can re-give meaning to spontaneous fermentation beer with the bacteria, where you have, for example, fruit, which is also left there to ferment to create bacterial contamination, so as to have live elements in our body. It means that we're moving in the direction of what Carlotta was saying. We must be very careful. We shouldn't be the super health, super clean, super everything, because otherwise we'll end up in another world, in other problems. So these would be beers that generate biodiversity. Yes, I think this is one of the very strong messages. I use it as a slogan. For me, live beer is exactly one of the points of communications that I use, and this is the case. Let's not filter, let's not pasteurize our product, and this is why we don't. The idea that the product must be alive and not dead. This is also true for wines. Wines that we normally drink, what are they like? There are two sides, two worlds to it. The ones which is opening today, which is a world linked to biodynamic and spontaneous fermentation, has opened a new trajectory in the world of wine to the cultural wave, which led to very interesting wines, is not necessary, but some of them are not necessarily live. The French were the first to do it. Yes, the French were good. They taught us how to use wine for a barrique and so on, and this made it possible to standardize the product and sell it well, while probably a more artisanal product is less standardized, but might be more interesting for the microbiota. So you can also do it respecting nature and I think you can do it with live products and maintaining recognizable and with some pleasing solutions. You don't necessarily have to do something difficult. Thank you. But to intervene on nature, we have to domesticate it and keep it our friend. Accompany it. Try to understand, well, respecting it. I think that's the most interesting thing. Maybe a question. Are there any questions from the audience? Yes, good evening. First of all, thank you very much for this conference and for all those who took part in it. I have a question which is for Dr. De Filippo.
Anche nel foglio che è stato dato
In
all'ingresso
the information that we were given at the entrance, we were talking about the extreme velocity or speed of adaptation of the gut microbiota. A change of diet led to, within 1 or 2 days, into a change of the gut microbiota. So if that is so, how is it that a lack of exposure at birth of the child to mother's bacteria can have some consequences that last at least up to 5 or 6?
Proprio perché questo contatto che è, diciamo,
Because this contact, which is a key contact, that is to say, these are the first bacteria. These are some bacteria that otherwise would not be seen, because it's not that otherwise these are bacteria, vaginal bacteria and fecal bacteria. So they're bacteria that you can only come across at that particular moment. These are bacteria that we're all born with, and our immune system has evolved to recognize those bacteria. If we don't have that sort of meeting with these bacteria, then clearly the flora will regenerate in future, but we have this initial lack. As far as diet is concerned, diet modifies very quickly. An experiment was done with volunteers where they were given a diet, either a meat diet, which was then shifted to a vegetarian diet, and within 48 hours there was a prevalence of some bacteria compared to others. This doesn't mean that you completely lose as if you were reshuffling the cards completely, but clearly diet favors the prevalence of some as opposed to others. And it is a system which is quite fast in answering, and because it is fast, to a degree we have to monitor it.
Innanzitutto faccio gli auguri per questa nuova stagione.
First of all, I would like to congratulate you and wish you all the best for this new season, thanking you, and yes, to everyone, including the people who are following. I would not appear to be use a word that starts with S and ends with a T. That is to say, as far as I've understood, but I already know it, most of our diet every day, living in Turin and so on and so forth, is we're eating S.T, while probably it's this S.shit that we should be eating. Can you give us some advice on bacteria and how we maintain our gut flora? Good advice to correct things and to rebalance our gut flora. What I told you, this doesn't mean that we have to eat what Burkina Faso's diet is. Certainly we have to rethink how to rebalance. We use too many refined foods and too many proteins and too many simple sugars. And so the fact that we have a balanced diet, like the Mediterranean diet, let's not look for particular things, but a good Mediterranean diet with the input of fermented food. I include, although we can't say it, also beer. Because if we could use fermented beverages, not too alcoholic, but bearers, as in the case of yeast, because otherwise we go to the chemist and we buy the Saccharomyces boulardii, which is a probiotic that is used, and possibly it might be better to introduce them in a more natural manner. And this is the message and the advice. Introduce and not lose contact with traditions.
Assolutamente, sì.
I wanted to say something. I don't want to seem senile, but there is to marry the wife and to use the ox of your own countries, which have been contradicted and favored. Contradicted because we talk about the great importance of biodiversity, but it is also true that we have, for thousands of years, that we have evolved with our food. The diet in Piedmont was the one in Burkina Faso and the one in China. And this had some common elements, that is to say that they contained microorganisms that entered our body that were different according to the places. In the past 50 years, we have seen the globalization of food and their standardization. So an important rule was, for example, food safety, which the European community has translated into an absolute control and sterilization of all we eat. This reassures us because it avoids salmonella and so on and so forth. But there is a serious problem. That is to say that we're once again having to start to input some, say, probiotics, because say you have a yogurt, then you take away something and then you add something to it. We discovering something essential which we'd forgotten. That is to say, when you go and look at the trend of autoimmune diseases, the development, the explosion of autoimmune diseases, but especially amongst children, this goes hand in hand with the standardization and globalization of food. There are some question marks that should be raised, and possibly a little bit of shit of the good one will have to start eating it. I agree. I'm very sorry that I'm politically incorrect by using the word, but that's it. There was a movie by Gavino, the Sardinian, who wrote the book Sardinian, when the sheep's stool fell into the milk, but you made cheat the same. Or the transplant of microbiota. But what about bacterial population? Is it typical of the person, a mind different from yours, for example? At what point can you generalize in terms of health and microbiota? There's certainly an individual variability, which must also be considered over time, because not only from person to person, but in the same person over time. For example, there is a seasonal variation, and it's also circadian. Yes, it changes according to the different parts of the day. You were asking me about standardization. Yes, to what extent can you generalize what is being said with all this advice? This advice, which is very precise, but on the different people may have different effects. The microbiota would appear in medicine one of the very personalized features as compared to others, because each one of us reacts in a different manner. The so-called probiotics, which are prescribed very often, have no effect on some people as compared to others, precisely because they're in a context which is different. You must always think of an ecosystem and therefore a balance of the various parts. And so there's still a long way to go. But yes, it's one of the possible these aspects because I would like to thank Paola Bonfante because she is the one who brought them here. Thank you very much, Paola. We'd like to thank this evening's speakers, Duccio Cavalieri, Carlotta De Filippo, and we would also like to thank Matteo Musso and Piero Bianucci. I will be seeing you next Thursday. Thank you.